Okay. Good. Yes.
Okay. Yeah.
Yeah.
Thanks a lot for the kind invitation. And of course, I'm very happy to talk about our recent work. And that deals with the fact that when healthy and sick cells are damaged or inflamed, then protein densities in the cell and the nucleus will oscillate over days. In particular, we have looked at p53 and NF-kappa B. These are very famous transcription factors. I come back to a little more in a second. And here you see a typical oscillating signal we look at. And I, of course, also come back to that. Now, what happens if you damage the cell? Of course, the DNA would like to be repaired. And our suggestion recently, and Jay knows this work quite well, he has heard a talk also a few weeks ago and so on, that we think it repairs on foci or droplets. And of course, I come back to that, how we think it works. If the cell is not repaired, the healthy response is it should die, which very, very often happens. But then if you have an internal oscillator in the cell, you could think about coupling it to an external oscillator. And we have worked quite a lot on that the last 5-6 years. Then you can get what's called Arnold tongues, something I worked on also in University of Chicago, and I'll show a little bit of that. And if you have Arnold tongues, you can have chaos, and then we have looked at how can that influence gene control and DNA repair, as I also just mentioned. Now, why do these transcription factors oscillate? It's because they are regulated by negative feedback loops. And I just show a few examples of that. And then I come to this important work recently that we have this foci formation around the DNA damage sites, and we think the repair will take place there. And we have a fantastic collaboration with Academia Sinica in Taipei. So then also we have looked at recently when two external oscillators couple to the internal oscillator, and what we, some way to our surprise, found that it enhanced the control of the genes. That's a collaboration with Peking and actually Shenzhen universities. And we have also worked on cells, and something going on right now from Harvard Medical School, that we really try to map out these Arnold tongues in single cells, and actually we have quite nice results there. So in the end, I will come to this. I already alluded to if deterministic chaos is relevant for gene production. And to our big surprise, we found that for some genes, indeed is. And then finally, something we work on really a lot these days and discuss these cases. If we go below oscillations, below what's called Hopf bifurcation, then you can have a what we call genetic resonance. And we actually think that we also are trying to get experimentalists involved in this project. So, okay, that's my outline. And I speak around 50 minutes, right? Yeah.
Yeah.
And you are welcome also to interrupt me on the way.
Yeah, sure.
Anyhow, I want to pay tribute to my late mentor, Leo Katanov. I think all this work I've done the last 10 years really started with a discussion with him 10 years ago, because as I already heard you collaborators, there are many, many oscillators in biology. And Leo and I discussed, we have had a lot of fun and a lot of good research on looking at oscillators in fluids. And then that was sort of initiated my work there. And I'll show you one example of what we did in the University of Chicago. But of course, as some of you already told me about 5 minutes ago, there are many, many cycles and clocks in biology. Cell cycle is very famous, and we just heard about circadian clock, which is of course 24 hours. Calcium oscillations in and out of cells are on seconds. Embryo oscillations is what's form our spinal cord. It's also have fairly long time scales, around 1 hour. Pacemaker cells, we all know, in hearts, and that's kind of also fast. And protein oscillations, in some sense, is a fairly new topic. I mean, the first experiment came a little more than 10 years ago after DNA damage, or if you inflame cells. But finally, also, lots of biologists look at population dynamics. And that's, of course, very slow time scale. That's on 1 year. You can have many. If people have looked at a piece of grass in Iceland, there are so many sheep this year. There are fewer next year. There are more next year again. There are fewer next year. So it also oscillates, and of course, due to interactions with other species. Quite interesting topic, actually. So there are many oscillators around us, and in the world. And but we have focused very much on. I wonder how do I get rid of this bar up here? Do you guys know? Okay. Anyway, if you do gamma radiation on human cells, MCS7, then p53 oscillates, and this fantastic collaboration we have the Galit Lahab group in Harvard Medical School, and here you see typical traces, and these. You can now have this running for, as I already said, for a week or so. Now it's not really a standard physics pendulum with exact same amplitude all the time. The amplitude varies a little bit. And why is that? Because in cells we have noise. And of course we take that into account in our computer simulations. But these are very typical traces. So the other work we do is with looking at alpha factor in yeast cells. And that's collaboration with Ping Wei in Beijing. And there you also see that NF-kappa B oscillate. And that has also been known for 10, 15 years by many groups. Actually, the first one to find it was actually David Baltimore in Caltech. So our research the last 5 years has been, can we control and understand it? And can we use it for gene regulation, control, and DNA repair? And the question to the answer to the last question is that we think we can. Now, these two transcription factors are extremely fundamental, and of course, they are the two probably most studied proteins in the world. Why? Because p53 is a cancer gene. It's mutated in nearly all cancers. Well, maybe only 50%. I don't quite know. And it's well known that people think that high value of p53 prevents chaos. And then you could say, why don't everybody has high p53? Well, it also induces apoptosis, so it induces aging, so it's a delicate balance. And it controls thousands of genes, and this number might be growing every day. NF-kappa B also controls, I say here 500 genes, it's certainly many more. And it's upregulated by inflammation, so if you inflame a cell, then you will see that NF-kappa B oscillates, and therefore it's of course very important in diabetes research. So it's not surprising that these two are probably the two transcription factors that are most studied both in biology, medicine, and also by insistence biology. But you guys probably know, but nevertheless, I show here that if we want a basic oscillator in a genetic system, then we need a negative feedback loop. Here I have a gene 1 that is positive, or gene 2, high production of 1 leads to high production of 2 if it's a transcription factor. However, you can also have this a negative effect, which you use the stacker here, positive, negative, and so on. And if you have a closed loop, it's like adding -1, right? Here's a -1, here's a -1, here's a -1. You should get a -1. If you get a +1, you have a positive feedback loop, and then you have a switch. You can either be in a high state or low state. Lots of people have studied that too. But actually, the most simple negative feedback loop in the genetic system is what's called the protein HES1. My cursor is jumping a little bit, I'm sorry. And I have actually studied that also recently. So HES1 is negative on itself. And that means that if you have high value of HES1, it goes to the DNA and it blocks its own gene. It's a little bit funny effect. And if you have then you can have a simple model with a time delay. And if the time delay is small, you have a steady state. If the time delay is a little bigger, you have a damped oscillation. And the time delay is slightly bigger, you have sustained oscillation. One of the famous negative feedback loops was introduced 23 years ago by Ilowitz and Leibler, called the repressor, and many people know this famous paper. They built in 3 repressing genes in E. coli. You see 3 expressing genes here, and indeed they did observe nice oscillations. Now we are more into what I call 4 here, that you have a transcription factor that produces another gene, another protein, but they bind in a complex, as I heard before, as we heard before by one of your students. It binds in a complex. If it binds in a complex, it's not free, and therefore it cannot produce. It cannot be transcription factor. So that causes the time delay very nicely, actually.
I see.
And also induces degradation, as I write here. So these are exactly the genetic network we have looked at, in top for p53 and bottom for NF-kappa B. And it's really a lot of work with my PhD student over the last, I would say, 10 years, and fantastic postdoc, Sandeep Krishna. I come back to him. So we try to map out the genetic network of here p53. And as we write here, typically p53 network is activated by irradiation, DNA damage, or by oncogenes. And you see all the proteins here. And okay, NF-kappa B, we also mapped out the genetic network. And how do you excite that? You can stress it, or you can, as I said, inflame the cell by trying to infect it with some external cytokine, and so on and so forth. Now, I come from the Niels Bohr Institute, where Niels Bohr is a great hero. And he also said, you should try to simplify to understand. and so we simplify them, and I show here, but I go back because I think I also told Jay before that I have realized this is a little bit controversial issue here, because biologists have told me, why do you want to simplify? I mean, here you have the full network and you know everything, you know much more if you want to simplify. And my answer is, we would really like to understand the basics of the oscillations. Why do these genetic network oscillate? We would like to understand that, therefore we simplify. And biologists, no, you should not simplify. And I say, well, here we have 25 variables and 100 parameters. We don't know how to do it, but anyhow, it's a little bit interesting story. I think in physics, we are trained, try to simplify to understand, then you can go back to the more complicated. Anyhow, that's what we did here is NF-kappa B. We just wrote a model with 3 coupled ordinary differential equation of NF-kappa B. And the so-called inhibitor it promotes, it's called I-kappa B. So we have measured the I-kappa B and I-kappa B protein, and they bind in the famous complex, lead to degradation. And that leads to these coupled, 3 coupled ordinary differential equations, which are extremely nonlinear. You see the degradations are quite nonlinear, and that's actually why it can be chaotic and so on and so forth. And you see here, it's very simple. That you have a complex formation here in the degradation of NF-kappa B. NF-kappa B binds as a dimer, sorry, and then there's a translation here. In principle, very simple. And we do exactly the same for p53. p53 promotes messenger MDM2 that translate to the protein and binds in the complex and cause degradation, as I write here. And so here we also have 3 coupled nonlinear ordinary differential equation. Fairly similar idea between these 2. Also binds as a dimer. It could bind more cooperativity, but whatever, and translate linearly. And we find beautiful oscillations with these simple models. That was kind of our projects here a little more than 10 years ago with Sandeep Krishna and myself and other people, a lot of students. And we could actually explain the first experimental data by Baltimore and White and others on NF-kappa B and also the p53 by this simple model. So, and I have worked quite a lot on what's called WINT, genetic network, which has to do with embryo segmentation, so-called somite formation. I don't have time to go into that today. Anyhow, so these are the model, and of course, me being trained in dynamical systems and chaos theory, like this kind of models, because we can also do a lot of analytic work in the sense that we can do a lot of find the fixed points, we can do stability analysis, we can see if there are Hoff bifurcations, we can see if there are period doubling, transition to chaos, and so on.
See.
That we can do with the reasonably low-dimensional models.
See. So,
okay, so
Mogens, when they make
yes,
complex P53 MDM2 or NF kappa B IKB alpha, their stability also changes. So the complex formation promote their degradation, or in your model?
Yes. It promotes the degradation, which actually you can see in these two terms. You see, these are the complex. These are the complex, and you have the minus on. So that is a completely important part of the degradation process.
Okay,
yes.
gotcha.
Thank you very much.
Good. Thank you. It's interesting.
Essential question, actually. Thank you. Okay, so now I will start first with P53, and then I come to NF kappa B at the end of the talk. Now, so what they do at Harvard Medical School, and Galit Lahav group is, I mean, sort of the pioneers there, because she started, I think, more or less 15 years ago, maybe even a little more, with Uwe Alon, and tried to do radiation on both sick cells and healthy cells. In some sense, we are most fond of the healthy cells, because we would like to see the healthy response, right? That you do DNA damage on healthy cells and see what happens. Of course, DNA damage on sick cells are also a very important issue, but they, as we unfortunately know, the sick cells, the cancer cells, are harder to go apoptotic. So that's, of course, fundamental issue here. But here I have a time series, and it seems to work. You see, P53 is green and MDM2 is red, and you see they oscillate up and down with a typical period of 5.5 hours. So it's fairly slow. And here you see a trace over 32 hours, a little more than kind of one and a half day or so. You also see with that, if you have a maximum in the P53, the maximum of MDM2 comes a little later, is phase shift, as it should be. As I also showed you in the traces we find, there is of course a time delay before MDM2 comes. Why? Because a P53 promotes MDM2, and so this is the right dynamical effect we can see here. So of course it has been a discussion, and I think 6 years ago in your conference, Jay, I was saying that probably the P53 oscillation have to do with the repair. It's likely, right? Because when you do first damage and the cell tries to repair, then the oscillations are very likely a part of the repair process. But at that time I didn't know how, and people asked me how. I said, I don't know, but it's very likely it's part of the repair process. Okay, so here I show you again our P53 model, just to show you. It's here you again, Jay, see the degradation term, the transcription and the translation term, and fairly simple. And you can get beautiful bifurcation. You can get beautiful oscillations. You get hop bifurcations with limit cycles in the usual way we like. And depending on the parameters, of course, you can induce more degradation, or you can induce more production, and so on and so forth. And you see here we have not so many parameters. We have 7, and some of them actually we know more or less the size of. So it's nice to work with. I think in this, at least precisely in this topic here, it's nice to work with fairly low dimensional models in order to understand. But of course, okay. So in various cells, people have studied actually quite recently, last year, like 2 years ago, that there's a rapid accumulation after damage. There's a rapid accumulation. We actually see the graph over here of P53 at the damage sites. I think I should have, here you see an accumulation of P53 at the damage site. Very fast, within 40 seconds, very fast. But it's also known that P53 promotes many other repair proteins at the damage sites, and people have measured that. Here, other proteins, you see, they grow after some time at the damage sites, also on the time scale of minutes. And you see also the movies here. So it's really something people have given a lot of thought in recent years. What happens at the damage sites? And you already see a very important point of our recent work, that there are some foci or droplet forms, right? Or there are droplets formed around the foci. You can see that over here.
Yeah.
You can also see it here. So there's clearly some inhomogeneity in where these both damage proteins and repair proteins are allocated in the nucleus. Now then there's a famous thing called Ostwald ripening. It goes a long way back to the famous Russian physicist Lipschitz, who worked many years with Landau, as I think we all know.
Lipschitz.
And at least us physicists know. And there's a very interesting, very basic phenomena. If you have a lot of droplets forming, they compete about the material. And there's a kind of critical radius. And since they compete about the material, if you're above the critical radius, these droplets would like to grow on the expense of the other ones that will tend to shrink. And that, of course, will cause that some will the winner takes it all, kind of, exactly here, that some tend to grow, and in the end only one will dominate the whole thing. One droplet will dominate the whole thing, because it will take all the material from the other droplets, and they will shrink, and in the end only one will win. So, and therefore there's of course in extreme space here is extremely important in this idea we have that the droplets compete, and how it's related to oscillations, because they are all the proteins diffuse around and are then allocated within these droplets. First we did a simulation, and this really worked with my fantastic collaborator, assistant professor here, Matthias Seltberg, and Alexander Luchetti, and the group in Taiwan of Shen. So we very well know that that, as I told you before, we start to do DNA damage, and we have oscillations of P53. And here we also show some numerical traces. Of course, with the model I just showed you, we can very, very easily find where we have oscillations. Now, if we now look at a situation where actually P53 is constant, then we find exactly that one of the droplets around the damage site will grow. This is shown on this. Sometimes my cursor is slow. I don't know why. It's shown here is the radius of the different droplets. You see in the beginning you have several, but the blue guy here will win, and the other guy, it will suppress the other droplets. As you see here, after some time they simply disappear, and one will win. And therefore, in that case, it's not optimal, right? Because why do I say this? Because if we believe that the repair takes place in the droplets, then if only one will win, then you will not have time to do the repair in the other droplets, and therefore there will be quite a lot of damage left. As we show over here. So actually show down here. Here you see the damage left. If you still look at the colors, there's 50% of the damage left in the orange droplet, which, as I show up here, quickly is disappearing, and the radius quickly goes to 0. Whereas the one that wins, well, the one that wins due to Ostwald ripening, here damage, all the damage will be repaired, and there's no damage left after some hours. So this is, of course, beginning to some DNA damage repair, but not optimal. Now I will show you a little bit other very recent just information for these droplet formations, because there's a famous paper by Piscina and al. Just also 3 years ago, also irradiation of cancer cell, and they found indeed that there are a lot of liquid droplets forming. They talked also about Oswald ripening, and actually, if you look here, you look at here as the radius of the biggest one, and here's time, and it's on log scale. There's some indication it will grow as the theory predict as t to minus 1/3. However, you also see indication, they actually didn't talk much about it, and we discussed it a lot. You see here, you see a lot of foci. But you actually also see after some time that the foci seem to disappear. Now, they did not measure p53, but there could certainly they see all the droplets forming around damage sites. They might also see that they seem to first to coarsen and then maybe disappear, and that might be due to the p53 oscillations, which they didn't measure.
I see. So, Mogens, so here the droplet is made by p53 or other things. So we don't know.
They also measure a few other proteins. I can't remember the names.
But it's
so
another
they measure
type.
There are many repair proteins. People know, I think, a lot of 200 repair proteins, and they tag some of them. And what I showed you a few slides before was that p53 is certainly high concentration around the damage site in the droplet, but also many other proteins.
I see.
And I think these shown here are some other proteins whose name I forgot. DP something. Yeah.
Okay.
Yes. So now we began to think it's. I keep saying that I have worked on oscillations for many years, and Matthias Heltberg, as Jay knows very well, was my PhD student, but then he went to Paris. So from me, he learned about oscillations, and in Paris, he learned about droplets. So we combined the two issues in the following sense. Because when p53 is high, here you see, by the way, oscillation of p53. When it's green, it's high p53, white, low p53, green, high p53, with a famous period of 5.5 hours. Then you see that the foci, they grow, the droplets grow, and p53 and other proteins are recruited down to the damage sites. However, then the level of p53 goes down quite low values, and then then they retract from the droplets, and therefore the radii of the droplet then shrink. So our idea was that for high p53 level, the droplet grow. For low p53 level, the droplet shrinks, and then p53 comes up again, and the droplet grows. So you see, our idea is that due to the oscillations of the p53, the repair droplet grow, shrink, grow, shrink. And that was exactly what I was missing in my, what I used to say, that it's part of the oscillation, part of the repair process. Why? Because then you prevent Oswald ripening. Then you prevent that one will dominate the whole thing. You will have droplets of more or less the same size, but you will not have that one will try to suppress all the others. Why? Because after these 5.5 hours, it has to retract in size again. And in the model we did, where we did a combination of least switch theory for droplet formation and oscillations, then you see that here are the radii of the droplets. It grows, it goes to 0. it grows, goes to 0. Well, one might still be slightly larger, as you see in our simulation made by Alessandra here, that one might still be bigger than the others, but it has to shrink again. That's the whole idea. So as I write here, oscillation helps for efficient DNA repair because it suppresses osmolar ripening. Now then, in our model, we looked a lot at, and we introduced some simple steps. We introduced actually a simple Markov process in the repair process of typically 100 steps. It's in some sense we don't know really, but we know, of course, there are some steps in the repair process, and that we had some time in that sense for the repair, and then we could see how much damage is left on the damaged sides. And what we found, to our big surprise, was that were interval actually fairly big, because you can see down here it's in logarithmic scale. So we actually go from minutes up to hours, but nevertheless there are a fairly big time scale interval where the repair is optimal compared to very, very short oscillation time or very slow oscillation time up here, where kind of the steps in the repair process doesn't, should I say, synchronize with the external oscillations. So we have a window here where actually, surprisingly enough, it's quite sharp window, but the window is quite big. That's of course a good news. But the best news is that the 5.5 hours is kind of here in the interval, although a little bit on the boundary of the interval, but it is in the interval. So the 5.5 hours is optimal for repair, but it certainly also, according to our theory, could be shorter. Actually, it could go down almost to 10 minutes. So yeah, so we had this theory last spring, and then I have just a simple abstract here of that we have the p53 oscillation that creates damage, and if we have the sustained oscillation, sustained value of p53, that means without oscillations, you have much more damage left than when you have oscillatory. And we have that these oscillations, as I showed here, suppress osmolar ripening. Then we had contact now for the last 2 years to a group of Sheng in Taiwan, Taipei. And actually Sheng, he is educated at Galit Lab in p53 oscillations. And he's building his own group now for last, I think, 1.5 years in Academia Sinica in Taipei. And they do a lot of very, very nice, actually can almost have them longer time oscillating that at Harvard. I don't quite know why. But they could for 6 cell lines, and here you have 6 cell lines. MCF7 is what they typically use at Harvard actually, and but then they also, and one of them is healthy, and I can't remember which one. I think it's RP1. I'm sorry, I forgot which one is healthy of these 6 cells. But then they could induce oscillatory p53 by damage. They actually do it chemically, or they could suppress the oscillation, which I think they do by a chemical called Nodlin. They could suppress oscillation, so you more or less have a constant p53 level that I show here, or you could have a oscillating p53 level here. That here you see for 2 different cell lines. Very nice oscillations over here, and more or less constant level. And then what they have a certain tag which actually kind of measure the damage level, called gamma H2AX. Not that I exactly know why it does that, but what is very, very important, in particular, you can see for this cell line here, is that after 24 hours, for the sustained level, constant level of p53, there's lots of damage left, whereas for the oscillatory p53, there's much less damage left. And that was really fantastic for our theory, that we got this experimental confirmation, so to say. And here you actually see the data, for instance, oscillatory versus sustained. It's within a biological measurement. Some of them are more clear than others, but there's always a complete clear trend that the oscillatory, you have much less damage after some time. Actually, you can maybe see it over here, what they did, maybe more clear, that if you look at number of damage site after I think the 24 hours and you do a histogram, you see that for the oscillatory it goes, you have very few left, whereas you have a broad distribution of damage site in the sustained P53. Okay. So now the last 10 minutes, I will just tell you what we have done quite a lot also in recent years and still do. And that was, I alluded to in the beginning, and this has been more in the NF-kappa B system, that you have typically you induce inflammation in also as well. This is typically actually healthy cells. Fibroblast, or what they do in Beijing is actually in yeast cells. You can put what's called tumor necrosis factor, and then you induce oscillations. And that is quite well known. Now, with Sandeep Krishna, we started almost 10 years ago. Why if we now oscillate this TNF from outside, can we then get the two to couple? And Leo Kadanoff found this fantastic picture that is 350 years old. It's by the famous Dutch physicist called Huygens. He actually was in bed with a fever, and there were two clocks on the wall. Actually, I think people say that Huygens invented the pendulum clock. But anyway, he found out, after some time they synchronized. So he built this ingenious experiment with his father, 2 chairs, piece of wood, and 2 clocks hanging there. And if the clocks are close, they synchronize. If the clocks are apart, they do not synchronize. As two metronomes on a piano. I think I jump over this. So when we have 2 coupled oscillators, and this I worked on during my PhD with my late supervisor Per Bach and Thomas Bohr, and then you have what's called Arnold tongues, and that has to do with the fact that when the internal frequency divided by the external frequency is close to a rational number, you get a resonance effect. So the Arnold tongue diagram, you have the external frequency on the x-axis and how strong they couple on the y-axis. And then you see there is an interval that here's the 1-to-1 Arnold tongue. We say there's an interval where you can pull the internal oscillator to the external oscillator. It's called entrainment. And you get that for all rational numbers, and you get these Arnold tongue widened. And it's a very interesting story. Then at some point they overlap. This is kind of green here, and at some point you might have chaos.
I see.
So I just want to show briefly, that's what actually we did in University of Chicago in the 80s, because if you have an experiment by Lipscher that you heat a convective fluid, you can have oscillations stable for weeks.
Okay.
And then when I came, I suggested, why don't we oscillate from outside? Okay. And then we had a conducting fluid, and indeed we found Arnold tongues to a very, very high precision in this experiment, and we could see bifurcations, we could see period doubling, we could see chaotic. So that's why I talked with Leo Kadanoff that we should do the same for the cells. And here I tell you this, now if we have an NF-kappa B model, I showed you in the beginning, and we drive by oscillating TNF, we get 5 coupled ordinary differential equation, very nonlinear, and other people have also looked at this, and we get the oscillating TNF in here and in here. And then what did we find? Well, we found beautiful Arnold tongues.
I see.
So here you see what the 1-to-1 Arnold tongue. So the external oscillator is the green, and the internal oscillator is the red, and they are completely synchronized. and they will be synchronized over an interval of the external green oscillator here. And if two tongues overlap, this I show here, then you can get either what we call 1/3 or 1/2. 1/3, the external makes 3 period, while the internal makes 1, and 1/2 is 2 to 1. And then the state you find depends on the initial point. So the initial point will tell you initial conditions if you go to one or the other. However, if you have noise, which we have in cells, it might jump back and forth between these two solutions. This is what we are going to see in the experiment. And I go a little bit fast here just to show the experiment. If many tongues overlap, we get beautiful chaotic attractor in this NF-kappa-B system. So I got contact to Savastai a few years ago. He was at that time in Zurich, and he had beautiful experiment on microstoves where you put the cells in a flow chamber. And if you have constant TNF, you have oscillations. So I talked with him, why don't you oscillate TNF through this microchannel? And if you see here, the oscillations are the kind of indicated by the gray here. They put in TNF and then it decays, put in TNF and it decays. And then amazingly, they found nice one-to-one entrainment. You can see here, one-to-one entrainment after some time. The different colors are different cells. Sometimes they were not entrained, as expected. So for me, it was amazing that you could see what we saw in fluids 30 years ago, you can see in single cells.
Yeah.
And here you see a typical biologist diagram on that. You see these very beautiful entrained systems sometimes, and not so entrained other times. You have around 200, well, 100 to 200 cells on the y-axis and time on the x-axis. And interestingly enough, they found that some genes produce more in entrained cases than when they are not entrained. Entrained is red and non-entrained is blue. So by having an external oscillator, you can actually change gene productions. Amazing. So we talked to him about, can you see overlap of tongues? And this we published a few years ago. And Savastai and Ryan Kellogg says very, very funny, because sometimes we see it oscillate in one state, it jumps to another state, it oscillates back to the first, it oscillates in another state. Oh, we said, this is exactly what we will call mode hopping, one state to the other, back to the first, that you can have, we know from theory, when two tongues overlap.
I see. So when they overlap, it's always hopping, or sometimes it's chaos, it's depending on...
Okay, I end by that, Jay. Thanks a lot for a great question. So Matthias Heltberg, he did stochastic simulations, and there are a fantastic technique called Gillespie algorithm, some of you might know. Instead of having Langevin noise, with Gillespie you do in terms of molecules, and you can control the noise simply by varying the volume, the number of molecules. The more molecules, the more deterministic, the fewer molecules, the more noise. It's fantastic technique. And what Matthias found, yes, if we put ourselves on overlapping of two tongues, it might indeed jump back and forth between the two states, and we had very nice agreement. I would say more on the qualitative level with the experiments. But you asked a great question, Jay, and I will end by that. If we have the external frequency here and the external amplitude of this TNF cytokine, well, if amplitude is low, we have one solution. I told you you could have an overlap of two tongues. Then you have two solutions in phase space here, you see, and it can jump back and forth. If you have many tongues, your question, Jay, if you have many tongues overlapping, we know from theory you should see chaos. And we did a very simple downstream gene model. We have oscillations of NF-kappa-B, is what you see here in the blue. Then we bind that with a certain cooperativity on a gene. Cooperativity you map by this Hill function here, right? This is called the Hill exponent. This number of molecules binding here, we have 3. And you have a certain affinity, how strong it binds, K here. So This binds to the DNA and gives a messenger RNA, and it translates to the protein. Very simple model, downstream model. What we found was if you have medium cooperativity and high affinity, the gene production goes down in the chaotic regime. If we have high cooperativity and low affinity, the gene production goes up in the chaotic regime. Very, very surprising. Very, very surprising. And if you see in the mode hopping regime, nothing kind of happens. It's more or less constant production, also in the single state. But so chaos is this is the model. This is model so far. But we have got a lot of biology labs interested in this. And of course, it takes some control how to get there, but I think we know. And so you can see here, chaos has a direct influence of gene control in that sense. I've just end now.
So when you have a high cooperativity with a low affinity, then with the chaotic dynamics induce the higher gene expression.
Yes.
Oh.
Exactly. That's exactly what I'm saying.
I
exactly
see.
what I'm saying. Higher gene expression, higher gene production. Amazingly enough, we understand it more or less. By the way, I mean, this paper here went into actually 50 news channels around the world. There's even a new YouTube movie about it, which we didn't do. Some people did for us. So anyway, you can search for that. I just want to end that we are now doing 2 oscillators with a group in Beijing, actually just moved to Shenzhen. And we just got this accepted in Cell Systems. And there we show that by 2 external oscillators, the Arnold tongue broadens. The Arnold tongue broadens, so you get more control. And I'm coming to an end here. And with Harvard, we are now trying to map out, the Arnold tongues, because first they do DNA damage, you have oscillations, and then they add an external oscillator called Notlin. It's well known. It's well known Notlin influences 53 loop by actually decreasing by actually binding to MDM2 and influencing the loop. But we see beautiful entrainment. We might even see pure doubling. Matthias thinks he sees chaos. I'm not sure, but we have now 4 Arnold tongues more or less mapped out. So we are just trying to finish this. This is something we call genetic resonance. I don't have time for that. And I will just end by saying that Cell Systems editors were very, very friendly to our recent papers, because actually they put fractals on the front page, Arnold tongues on the front page, because the editor said, oh, it's very beautiful because it looks like sunrise, and even chaotic signals on the front page.
I see.
And therefore, I will end by thanking my collaborators, very important in this project. Leo Katanov was my late mentor. I was in Chicago for more or less 4 years and went back every year, and we discussed a lot. I miss him a lot. And Harvard is Harvard Medical School, and the way Beijing, Shenzhen, and Shenzhen is Taiwan, and Sandy Krishna. And then I have a fantastic little group now, Matthias Helberg, Alexander Luchetti, you know them both.
Yeah.
Jay. You probably also know Kim Sneppen.
Yeah.
And here are some of our results, and thanks to our funding. So thank you very much. I think it was exactly 50 minutes.
Yeah, perfect. So let's start.